Good morning, everybody. All right, today is your chance to pepper me with questions about anything whatsoever to do with the material that we're going to be covering in the exam. Ah. I'm just going to sit here, drinking my coffee, staring at my screen, thinking thoughts till I get some questions. All right, Sophia, yep, that's right. There will be 40 questions. They will be, as best as I can, spread out pretty evenly over the 4 PowerPoints that we have covered, which are the genome structure, the transposons PowerPoint, gene expression and transcription, and then RNA processing and translation. But as you can remember, there's an awful lot of stuff in there. So there's plenty of material for questions. Also, don't forget too that there'll be quite a few multiple answer questions, but how can I put it? Essentially, there's a negative penalty for the wrong ones. And so for the multiple answer questions, don't just tick all of them, because if you do, you'll end up with a lower score for that question than you would if you just picked the correct answers. All of them might be right. Only 2 of them might be right. You just don't know. You have to pick the ones that you think are right. Now, you can't end up getting a negative score for that question, but you can end up getting potentially a score of 0, even if you get one of the answers right, if you pick multiple wrong answers. So yeah, it'll be pretty much the same, similar level of difficulty, more or less. There'll be some working out of stuff, particularly things involved in transcription and then translation, right? So it's not going to be just fact-based. You're going to have to kind of figure things out, right? Based on, the information that you've been given in the PowerPoints. Okay, anyone got any questions? Questions? What are your questions? Spliceosomes. Good question. So spliceosomes are they similar to ribosomes? No, they're really very different. So in some senses, they're similar because they consist of RNA and protein, multiple proteins, right? But I mean, I wonder if there are other proteins that are similar. There must be, but I'm not fully aware of them. So that's a similarity, yes. But in terms of what they do, very, very different, right? So think about it one way. All right, here's a question back. What is the substrate in a ribosome? Kind of couple of different ways you can answer that question. Right, if the ribosome is an enzyme, which it essentially is in many ways, what is its substrate? What is the thing that it catalyzes? What's the end product that you get out of a ribosome once it's done its job? Ribozymes. Well, ribozymes is what, good question. Ribozymes are what do the work in ribosomes. And Rogelio, RNA is, I guess you could call that a substrate in a sense, right? And that's why there's probably a couple of different answers to this. RNA is the template, right? So you're not modifying the RNA in any way, if you think about it that way, right? So the RNA that goes through a ribosome comes out the other end, essentially, unchanged, right? There's no difference from when the ribosome begins to when the ribosome ends its job. So you're all kind of on the right track. What enzymatic reaction do the ribozymes in ribosomes catalyze? What things change? Think about it that way. Good question. This is fun stuff. What changes as a result of a ribosome's activity? Not the RNA, or the mRNA rather, or even the ribosomal RNA. That stays pretty much the same. What changes? No, transcript's another word for the mRNA. So same kind of answer. Doesn't change. Now, if we're talking about RNA polymerase, yeah, I mean, RNA does change, right? Transcript does change from when an RNA polymerase starts to when it finishes, because that's how you get the mRNA. All right, so can I go further along? What reaction does the RNA polymerase catalyze? Kind of getting you thinking about this in a different way. That's the aim. What reaction does the RNA polymerase catalyze? What does it do? What is its purpose? And Sophia, we'll get back to, yeah, change DNA to RNA. But so, okay, let's put it another way. So this is kind of fun. When you transcribe a gene, when you express a gene, do you take that piece of DNA in your genome and turn it into RNA? Yes or no? So once that gene is transcribed, now you have RNA in your genome, in your chromosome. Is that true? No. So the DNA stays the same. It never changes, essentially. Well, I mean, it kind of does a little bit, but in other ways. So you're not changing the DNA to RNA. What are you doing with RNA polymerase? You can think of this in the way we call them a transcript, right? So all of you have a transcript, right? Official or unofficial or whatever. That transcript is a copy of all of the grade courses you've taken and the grades you got for each course, right? I mean, you're kind of professional students. You kind of know this stuff better than I do, really. So if your transcript is a copy of the records held by the registrar, presumably, what is RNA? Anyone? It's not an original copy. It is a copy, right? The original is DNA, essentially, right? That's the kind of, that's the version that is stored away in like the registrar's computer, or filing cabinet or whatever, right? The transcript is a copy of that, and you can make multiple copies, right? Just the same as when you get your transcript, you can go take it to a photocopier and print off 100 copies if you wanted to. Not sure why you do that, maybe for like job applications or something, right? So RNA is a copy of DNA, right? In a different form because it's RNA rather than DNA. Okay, so kind of winding back a little bit. If RNA polymerase makes RNA, what does RNA consist of? An mRNA transcript, what does it consist of? You'll see where I'm going in a little bit. What are the individual things called that make up your strand? No, not amino acids. That's protein, Sophia. RNA is a ribonucleic acid, right? No, polymerases are what make those large macromolecules. They're nucleotides, right? RNA nucleotides. Those are the bases, the AU, Cs, and Gs that make up RNA. So what holds those bases together? What bond joins those together that RNA polymerase makes? What is that bond called? Begins with a PH. Yeah. Check my spelling. What's the bond? Yeah, good stuff, Sophia. Phosphodiester bonds, right? That's the name of the bond that joins together bases in DNA or RNA, right? So you could say that RNA polymerase, what it does is it catalyzes the formation of the phosphodiester bond, right? Good. So rolling back to ribosomes, what does the ribosome or the ribozyme, right, in the ribosome, what does it catalyze? Formation of what bond? So what does the ribosome put together in sequence? You already mentioned it once further up above when you were talking about RNA. 2 words. First word, 3 syllables. No, that ribosomes are what make those bonds. What comes out of a ribosome? mRNA and what? What else? Amino acids. Yeah, kind of, Jimena. Kind of. Right? So actually, amino acids are the individual bits that go into ribosomes, similar to nucleotides, individual bases that go into RNA polymerase. What happens when you join a series of amino acids together? What is that now called? What's the name for that? You're on the right track. Yeah, the protein, peptide bond formation, Sophia, you got it. Right, so ribosomes catalyze the formation of a peptide bond between, as Jimena put it, amino acids, right? And that, after a while, Rogelio, generates the protein, right? And so protein is really the kind of the final functional version of what you get from gene expression, right? It's kind of like the three-dimensionally folded, funky-looking object which does something, right? The polypeptide is the string of amino acids in series joined together by peptide bonds that once it's finished and folded makes the protein, right? Awesome. Okay, so on another thing that ribosomes do, here's another kind of little wrinkle to think about. What brings in the amino acids? How do you translate that mRNA sequence to a series of amino acids in a chain or peptides in a chain? They're called peptides once they end up in a larger thing. What brings those amino acids in to be added to the polypeptide chain? Think about it. Begins with T, which I should drink more of because I'm British, but I don't. There you go. There's another way to think about it. Maybe you remember that way. I actually drank so much tea growing up as a child that it's just put me off tea for the rest of my life. Even the dog had a cup of tea every day. Can you believe that? It's kind of a little bit ridiculous. But anyway. Yeah, thank you, Sophia. tRNA. So T stands for transfer because it's transferring the amino acid to the polypeptide chain. But you can also think of it as translate as well. So tRNA is translating the series of words in mRNA into a series of amino acids in the polypeptide chain. Okay, so that, here's another question. What adds the amino acids to the tRNA before they enter the ribosome? Has to be something else. What's that called? Long, awkwardly to pronounce word series of words. This is probably one that you just need to look up. I mean, you can try and remember it too, but what's the enzyme that adds the amino acid to the tRNA? I promise we'll get back to spliceosome, Sophia. Almost there. What enzyme adds amino acids or an amino acid? No, not ligase. Ligase joins together bits of nucleic acid. So breaks in DNA or, typically just breaks in DNA. So not ligase. So made up of, at least two words. I'll give you a clue. The first part of the first word is amino. Ooh, almost. If you get rid of the ation at the end, it's amino acyl. Yeah. What's the next word? No. Well, yeah, kind of. Amino acyl tRNA, and then the next word. So kind of three words. Probably not helpful, am I? How could you turn the word? Yeah, synthetase. So he's kind of synthesizing, asing, right? Whenever you have A's, thank you, Azucena, that means it's an enzyme, right? So it's synthetase, protein asing an amino acyl group, right? So the amino bit is the amino acid that's getting added onto the tRNA. So it's amino acyl tRNA synthetase. There you go. Mouthful for sure, right? So don't go trying to say that once you've had a couple of, I don't know, Sprites or something. So where was I at? Yeah, okay. So now we have an amino acid stuck on a squiggly thing called a tRNA. What comes out of a ribosome after, how can we put this? What do you do before you make the peptide bond? So peptidyl transferase, the ribozyme that Sophia mentioned earlier, right? It does two things. The second thing is the formation of a peptide bond. What's the first thing? And it does this to the tRNA in the P site, the middle site of the ribosome. So don't forget, these amino acids are covalently bonded to the tRNA. They don't want to come off, at least not easily. So how are you going to stick things together if they remain stuck to tRNAs? You just have like a protein with a whole load of tRNAs dangling off of it, which would just look silly. You'd run out of tRNAs essentially. So what's the first thing that you have to do before you can form a peptide bond? tRNAs in there, right? So you've got two tRNAs, one in the P site and one in the A site. What do you do to the amino acid that's in the P site or the polypeptide chain that's in the P site? What's the first thing that you do? Yeah, good job, good stuff, Aurelio. It removes the amino acid from the tRNA, or if translation has proceeded a while, the polypeptide chain, right? So the peptidyl transferase does two things. One, it removes the amino acid from the tRNA, the squiggly thing, right? That's in the P site. And two, it sticks it to the other amino acid that's attached to the tRNA in the A site. Awesome. So the ribosome, its substrate or substrates, essentially, if you think about it, are the tRNAs with amino acids and amino acids to form peptide bonds. Okay, all good on that? Cool. So don't forget, adding an amino acid is not just about those two functions in the ribosome. It's also about adding an amino acid to the tRNA, right? Because if you don't have tRNAs with amino acids on them, you don't get protein, right? You don't get that whole translation. So that essentially, to put one amino acid onto a polypeptide chain, you have to A, add that amino acid to the tRNA, and B, that tRNA has to go into the ribosome and have essentially the polypeptide chain stuck onto it. Okay, so remember the kind of the whole thing. Okay, so now we're going to get all the way back to Sophia's original question of the spliceosome. Okay, so if in ribosomes, the substrate are tRNAs with amino acids and essentially amino acids for peptide bond formation, what's the substrate of the spliceosome? What does the spliceosome change? What is being spliced, right? Or changed from? It removes introns, right? Introns are present in what? You're 100% correct. Where would you find introns in terms of RNA? What would that thing be called? So there's non-coding regions of RNA. Yes, that is 100% correct. So when you have a eukaryote, ooh, dang it, I already told you, it's eukaryotes, right? So when you have a eukaryotic gene and you transcribe it into mRNA, is that a mature RNA? Yes or no? Is that ready? No, it's not. It's called, well, if it was mature, you wouldn't need to take the introns out of it, right? So what's the name of that molecule before it's been processed? It's not mRNA, it's, yay, thank you, Jelani, pre-mRNA or precursor mRNA. Well, you could even think of it as like pre as in before, right? Is another perfectly acceptable way of thinking about it. Okay, so pre-mRNA or precursor mRNA is the substrate for the spliceosome. Okay, and it removes introns. How does it do that? How does the spliceosome remove an intron? What are the steps? Don't worry about the order. We can put them in order afterwards. But what things need to happen? There's 3 or 4, I think. 4 different steps. Is it just like pizzazz, magic that happens? Or are there a series of things that have to happen in order? It'd be nice to think of it as magic, but unfortunately, that's not very good science. That's what happens when you design Apple products. Magic. Not that I'm biased against Apple or anything. Okay, recognized as a splice acceptor site. How are those defined? What do those look like in terms of sequence? There's one at the 5' end of the intron. That's correct. Is there one at the 3' end of the intron? Okay, so there are 2 splice acceptor sites. What sequence do they have? Conserved sequence, at least. What is recognized by the spliceosome? Or the individual parts, at least. Okay, GUAG or GUCAG. I bet GUAG would be fine. Okay, and there's another highly conserved invariant base. It's about halfway, well, not really halfway, it's about kind of like that far up from the 3' end. What is that called? What's its purpose? So you got 2 out of the 3 conserved regions in introns. The rest of introns is like, could be anything. Yeah, it's an adenine, right? It's called the branch point. And that's really important because that's how we form the lariat loop, right? Just remember cowboys, woohoo, yeehaw, like stuff, I guess. Okay, so GU, brr, A, brr, AG, right, are the bases that are recognized. So we have to recognize those. So we recognize them. What happens next? If you think about splicing an old-timey like film reel, what do you have to do to get something out if you're an editor, let's say? Marlon Brando or, you know, any of those famous people. Was he an actor or a director? Martin Scorsese, there you go. That's what I was thinking of. Godfather, stuff like that. What do you have to do to the pre-RNA? pre-mRNA, once you've recognized that intron, what do you do then? Yes, but you're missing a step. So how can you think about, I have a piece of string, right? And from the piece in the middle, you want to create a loop. How can you do that if you have an intact piece of string? You can't, right? So what's the first thing that you have to do after you recognize that? You have to cut it. Yeah, exactly. And that's always at the 5' end. And then when you take that end and you bend it back and you stick it to the adenine to make the loop. So you just skipped a little step there, that's all. Cool, okay. So we now have exon 1, or the upstream exon. We have a lariat loop, but that loop is still stuck to the second exon. What do we need to do now? Essentially, you could just repeat your answer. You just need to tell me which end of the intron. You've already cut the 5' end and stuck it to that branch point. Cut the 3' end. Good. So once you've done that, what's left? So now you have 3 pieces. First exon, or the upstream exon, the lariat loop, and the downstream exon. What do you need to do now? Yeah, that's right, Rogelio. You need to stick them together. And that's it. That's what splicing is. That's what the spliceosome does. And there's a whole bunch of kind of RNA and proteins involved. Actually, can anybody remember what they're called? You pronounce it the same as you would schnapps, but it sounds weirder. If anyone's ever had schnapps, it's very nice. Anybody remember what those things are called? They've got like this really long, horrible name. Begins with a sn. No, that's all right. They're called schnurps, hence schnapps, which stands for small nuclear ribonuclear proteins. Yeah. So that's the individual parts that make up the spliceosome. They are schnurps, small nuclear ribonuclear proteins. There. And ribonuclear protein means that they're RNA and protein, right? So they have that in common with ribosomes, and the ribosomes are RNA and protein, but they do very, very different things, very different substrates as well. Ta-da! Cool. That was an awesome question, Sophia. Thank you. Appreciate that. Okay, more questions. Hey, yeah. These are always fun, kind of getting questions like that, because then they lead to more questions and more questions and more questions. And then at the end of it, hopefully you now understand things kind of in a different way or better than you did when we started, which is the point. Okay, what else? Okay, so how about we go back to translation a little bit? What does the tRNA use to recognize the correct codon? What feature of the tRNA is used to recognize the codon in the mRNA? What's it called? So if you have codon, you have a, yeah, anticodon. I've always tempted to make like a bad joke, but it involves religion. I'm always a little wary of doing that. But yeah, it's a codon and the anticodon, right? So if you have, here you go, it's another way to think about it. If you have, oh, there you go, another question. What's the start codon? What 3 letters does that have? You're 100% correct, Rogelio. But moving on to the next question, what 3 letters does the start codon have? AUG. Good. What's the anticodon in the tRNA that recognizes an AUG? Who's calling me at this time? Ugh. Someone's selling me a extended warranty on my car or something like that. Bleh. Always from Pennsylvania for some reason. I don't know if like they're the spam call center of the US or something. All right, so what's the anticodon for AUG? So the tRNA that recognizes AUG, what is its anticodon sequence? If it's the anticodon, key is the word anti. What's the opposite to AUG? Ooh, almost. But what's that T doing there? I'm waiting. UAC, that's it. Good stuff. Right. So remember, there's no T's in RNA, right? It's in DNA. Okay, so it's UAC. What about, ooh, let's think up another one. Doopy-doopy-doo. Get the codon table up because I just can't remember what's what. Oh, before we move further on, what amino acid will that tRNA with UAC as an anticodon, what amino acid will that tRNA be carrying for the start codon? And this is the same in all organisms, anywhere. Prokaryote, eukaryote, doesn't matter. What's the amino acid that's going to be attached to that tRNA? I think you can even, I'm not a big sports fan, but I think there's a baseball team. Is it in New York? Yeah, the Mets. I don't know what city they're from. Could be San Francisco. Who knows? Anyway, methionine, right, is the one that is on that, right? So hang on. Yeah. Let me go over here. Okay, so going to the codon table that's in slide 38, right, of PowerPoint 16. That's, what about, if you had ACA in your mRNA? What would be the anticodon on the tRNA and what amino acid would that tRNA have? Okay, UGU, good stuff. Now, what amino acid is it going to have? So the codon, Sophia, is ACA. So the codon is the one you need to look up in the codon table, not the anticodon. So I can see the mistake you made, and that's fine, but let's clear that up. What's the codon? What does ACA encode in terms of amino acids? Exactly. So that encodes a threonine. Right, the reason it's not cysteine is because the anticodon is not the information that encodes the amino acid, it's the codon. The anticodon is really the means by which the correct tRNA recognizes that codon. Okay. Just a quick question. What bonds hold together the tRNA and the mRNA? Same as the ones that hold together the two strands of DNA? Nope. Phosphodiester bonds are what hold together the individual bases in the tRNA and the individual bases in the mRNA. Hydrogen bonds, that's it. It's an easy mistake to make, generally, but the reason why it's hydrogen bonds is those tRNAs have to leave, right? And so you want them to come in like stick, like Velcro, and then unstick them when you need to kick them out, right? The back door, essentially, the exit side. Okay, so this is something that is a kind of a handy skill. Okay, let's give you just one more just to practice. Oops. Okay, anticodon and amino acid, please. Correct. And oops, crab sticks. I went to the wrong. I'm pretty sure that's correct. Let me just double check. Yes. Excellent. Good. I'm glad you're getting that. I'm really happy about that. So just the final wrinkle or the fly in the ointment, so to speak, does that codon, does that anticodon or that tRNA have to recognize all three of those bases of the codon GGG? And I chose glycine specifically to illustrate this. Does that tRNA? No. Okay. What bases are really important of those three? They're not all equal. Which bases of a codon matter the most? Well, which end? There's two ends, right? The star or the other end? The third base end matters the least than the other. Yeah. So the first two bases, which in this case is CC, I mean, they're all C, but that doesn't, yeah, they're all C. No, G, dumbass. Okay, sorry, shouting myself. The GG matter. The third G doesn't matter as much. So the third G is less important, and particularly for glycine. Now, if I show you the codon table, okay, so if instead of recognizing GGG, that tRNA recognizes GGC, is that going to change what amino acid should be there? No, exactly. Because that's right, because the third is least important. Now this varies, right? So don't take it as a given that the third base is just not important at all, right? If you look at methionine or tryptophan, UGG, UGG, it's like UGG boots, right? it's very, very important that only those 3 bases are recognized. Because if not, then you get all kinds of problems, right? If that third base in the tryptophan codon up here on the top right, UGG, wasn't important, then you could potentially end up with a stop or a cysteine instead. But with glycine, it doesn't matter what that third base is, because all 4 of those codons, or 4 of those third base letters, equals glycine, right? That's called wobble. So the degeneracy part is the fact that you can have more than one codon encoding the same amino acid. But that doesn't always mean that it's just the third base that differs, right? So if you look at leucine up here, the first base also differs, right? So this isn't an issue of wobble, this is just called degeneracy, right? But for ones where you have like the first 2 bases the same for all the codons that encode that amino acid, the third base generally doesn't matter. So in a tRNA doesn't have to be specific for glycine. All it has to do is recognize GG, and that's it. The rest doesn't matter for glycine, right? So this is much more conditional than kind of a simple 1 tRNA, 1 codon deal. For some tRNAs, very, very important. Methionine, for example. For other tRNAs, not important at all. For others in turn, right? So if you look up here at tyrosine, it's kind of important. It's like halfway in between, right? So it's either U or a C is great. Not an A or a G, that's catastrophic, right? Then you just wouldn't get a translation stopping. You'd just have a tyrosine there instead. Okay, so wobble is a much more kind of like handshaky kind of deal. You actually just have to look at the codon table and see whether or not that third base is important and how important it is, right? Generally speaking, it is less important, but that doesn't mean it's not important, right? Okay. Anybody have any last minute questions? That was super cool. Always takes a little bit of time to get going, so don't worry about that. Okay, so there's lots of details, but those details I'm teaching you not because I want to just like make your lives miserable when it comes to studying an exam. It's because without those details, the bigger things don't make sense. Right? You have to understand those details, because otherwise it's just like a bit of a, ooh, yeah, this is the process, right? You're not going to be able to actually understand what's going on. So for molecular biology, you can't take the big picture apart from the little picture, essentially. You have to be able to do all of it, because it all works together, essentially. All right, everybody. Ladies and gents, thank you very much for your time. That was a lot of fun. And some of you I'll see later today. The rest, I will be in Zoom on Friday morning for probably about half hour or so, I guess, in case you have any last minute questions or issues with the exam. If not, the exam will, by magic, it's like Professor Magic, appear at 9 a.m. and I will make sure I get the time right and the date and everything. I'll try and get it all right this time, because there's always some bloody button that I don't press in Blackboard. But your exam will appear at 9 a.m. and it will be available until midnight Friday night. If you have any questions throughout the day, just send me an email. I'm going to be in some meetings and stuff, so I might be able to reply immediately, but I will reply as quickly as I can. All right. Thanks very much, everybody. Catch you later. Good luck in the exam.